Fun science, fun science, fun science, oh yeah! Now we'll be talking about proteins. Proteins are macromolecules found in many cells, and they have many different functions. They play roles as structural elements, enzymes, and signal receptors. Proteins are essentially polypeptide chains, and these chains are made up of amino acids. Amino acids have a general form like this. You have an amine group right here called the N-terminus, an R group which varies between the 20 different amino acids dictating their structure and their function, and you have your C-terminus, the carboxylic acid right here. Now as you can see, it's an amino acid, which is an ideal term because it is a diprotic acid. You have a hydrogen on this side and a hydrogen on this side, both which can be deprotonated. The hydrogen atom on this side, for the amine group, has a pKa of approximately 10. And the hydrogen of the carboxylic acid has a pKa of approximately 2. Given this characteristic of amino acids, they make for ideal diprotic acids in buffering solutions.
So if you want to have a way to visualize this more, you can put it on an alpha fraction graph. Now what this does, here's a generalized version of it. You put alpha fraction on your y-axis and pH on your x-axis. What this does is it shows the 3 different protonation forms. Here you have your fully protonated at low pHs, your fully deprotonated at high pHs, and in the middle you have your neutral. So here, showing what in our basic proteins, here you have the fully protonated form. As Bethany said, on the amine group you have another hydrogen there, and that makes it a positive charge. Over here, your carboxylic acid also has a hydrogen, but that makes it neutral. So the full charge of this protein would be positive. Over here, to the negative one, at a low pH, which doesn't have very many hydrogens available, you lost your hydrogen off of your amine, which means that it's neutral, but you also lost your hydrogen off of your carboxylic acid group, making it negative. So this has a net negative charge. Now look at not high and not low pHs, but neutral. You have your neutral form. But if you look at it, it's actually called a zwitterion because it has both a positive and a negative charge. You have one hydrogen on your amine group, which makes the nitrogen positive, but you lost your hydrogen off of your carboxylic acid, which makes that negative. Overall, it's neutral, but again, it's a zwitterion because it has both positive and negative charges. Your alpha fraction graph also gives you more information. If you do not already know the pKa's of your hydrogens, you would also be able to get that from your isobestic points. What an isobestic point is when your 2 lines cross over each other. So right here and right here. Your isobestic point is equal to your pKa. So when you look at this, you should estimate that the pKa here is about 3, looking at our beautiful axes, and over here it is about 8. That's about what Bethany said the pKa should be, around 2 and 10, plus or minus whatever units based on your R group.
There you go. Okay. Now we'll be talking a little bit about buffers. Buffers are essentially solutions that are resistant to change in pH. They're made of weak acids and weak bases. When you add an acid or a base to a buffer solution, that acid or base reacts with the other acid or base, basically negating any added H plus ions or OH minus that was added to the solution. This is incredibly convenient when you're in a lab because then you can control the pH of your solution so that certain reactions can or cannot happen. An equation that helps us describe a buffer is the Henderson-Hasselbalch equation. In this equation, pH is equal to the pKa plus the log of the base over acid. Now, the base over acid is a ratio, and therefore you can either use the amount of moles of base and acid, or you can use the concentrations of base and acid, as long as it's kept consistent. When choosing the proper buffer system for a reaction that you want, you first look at the pH range that you want to stay in. So let's say that you want a pH of 4 for your buffering, or for your total solution. That means that you want a pKa for your buffer that is within 1 unit, so -1 or +1 unit of 4. And then you take that pKa and you add it to the ratio, or the log of the ratio of the base to acid. This helps you determine how much base and how much acid you want to put into your solution to make your specific buffer. Hey, Ingrid, how do these buffers work?
Glad you asked. You can look at a titration curve to see an example of this better. On the y-axis, you have your pH, and on your x-axis, you have the volume of acid or base that you would add. Here, we have base, because as you see, the pH goes up. So, here is called buffering region. As you can see, the pH doesn't really change, even though you're adding a significant amount of base to your system. This is going to be known as your buffering capacity. Although, when you get to about however much volume is added right here, you start to see a significant change in your pH. Right here is known as your equivalence point. At this point, you have about a 1-to-1 ratio of your protonated form, that will be right here, and your deprotonated form of your acid. As you can see, again looking at the example, you have a hydrogen on both the amine group and the carboxylic acid group, as opposed to here you only have a hydrogen on your amine group, but you have your negative charge on your carboxylic acid group. So, here's your buffering capacity, and here is where it starts to change. Of course, this is a diprotic system, so we have a whole other form to go over here. Again, looking over here, you have another buffering region that the pH doesn't really change until you get to this point, where you have your second equivalence point between your singly protonated form with still a hydrogen on your amine group, and over here you have your fully deprotonated form, which has a negative charge. So if you look carefully, the amount of titrant added between your first equivalence point and your second equivalence point is equal, because ideally you will have the same amount of your neutral's Witter ion to react with all the base that you add as you had of your fully protonated form. This is known as the formal concentration, which we should have already covered in class. So looking here, you can also get information about the pKa. Again, 1/2 of your equivalence point, which would be right here, is also equal, the pH at this point is going to be your pKa. Also, if you want to look at your second one, halfway between your first equivalence point and your second equivalence point, right here, is going to be your second pKa. You can see for this system we have about a first pKa of 2, and here it looks to be about 7. Again, that fits because this is a protein, and those are the approximate pKas.
Now it's time for our case study. We're going to talk a little bit more about the bicarbonate buffer system. As we know from class, this is a buffer system used in our blood, which at a physiological pH is 7.4, roughly. This buffer system is displayed below. We have CO2 in its gaseous form, CO2 in its dissolved form, dissolved in our blood, and then with water, creates carbonate, H2CO3, and then hydrogen ions and bicarbonate, H3O3 minus. Now we talked a little bit about acidosis and alkalosis in class, but now we want to apply it to a real-world situation, such as overdosing on aspirin. There's a component in aspirin that makes us respire more. In other words, it makes us hyperventilate. Now when we hyperventilate, we're breathing in an awful lot of oxygen, but we're also breathing out an awful lot of CO2. It's this breathing out of CO2 that forces this entire system to shift this way, because our body needs to be able to make the CO2 and basically put it into our lungs so that we can expel it. When it does this, it uses up all of the H+ ions in our blood so that we can make the CO2, and by doing that, severely decreases the concentration of H+ ions, creating alkalosis. Our blood basically becomes too basic. When our blood becomes too basic, the proteins that are present in our blood begin to denature. As we saw before with our titration curves and our alpha fractions, and a little bit about proteins that we know that once the proteins are deprotonated, they'll change their shape, and therefore they'll change their function, no longer working how they're supposed to work. So when our blood becomes too basic, and we start suffering from alkalosis, we die because the proteins in our blood are no longer working, and all those physiological processes are slowed down or completely stopped. Pleasant. Fucking A. I'm getting so nervous that I can't talk. Fuck.
Ready?
Yeah.
Yeah.